Hello everyone. Today we will discuss one of the advanced technique, that is flow cytometry. So when it comes to analyzing cells in a lab setting, flow cytometry is widely used and it is a comprehensive single cell analysis method, and we can utilize this as an important technique not only in cell biology, but we can utilize this technique as it is in immunology and molecular biology. So let us discuss principal instrumentation details and applications of flow cytometry. Now, before going into the depth of this knowledge, I would like to emphasize on terminology. So first of all, let us understand the meaning of the word cytometry and flow cytometry. Cytometry is measuring physical and chemical properties of single cell, but flow cytometry measuring physical and chemical properties of single cells in a fluid stream in instrument called as flow cytometer. This is the broad principle of flow cytometry. It is a technology that simultaneously measures and then analyzes multiple physical characteristics of single particles, usually cells, as they flow in a fluid stream through a beam of light. The properties measured include a particle's relative size, relative granularity or internal complexity, and relative fluorescence intensity. These characteristics are determined using an optical to electronic coupling system that records how the cell or particle scatters incident laser light and emits fluorescence. So this is the principle. So if you want to study an individual cell, or even you want to distinguish a particular cell from the other cell, then you have 3 to 4 different criteria. That is size or shape, then metabolic reactions, density, receptors on cell surface. So there are large number of cells having different types of shapes. Then you can have different types of receptors on cell surface, and that also can be a criteria to distinguish one cell from other cell. And then in different types of cells may also have differential metabolic behavior or metabolic reactions also. So basic pathway may remain the same in different types of cell, but all the other types of cells which are responsible for giving them the functional outcome may be different in different types of cell. So all these 4 criteria can be exploited in a flow based assay where we actually utilize these 4 parameters to distinguish one cell from other cells. Now let us have look at these mammalian cells with different sizes. For example, sperm cells having size of 3.86 micrometer, RBCs, that is red blood cells, have 5.76 micrometer in diameter. So in terms of size, different mammalian cells are different from each other. Similarly, cells are different in terms of the density as well as the metabolic reactions and cell surface receptors present on the cells. So for analyzing these cells, you are looking for a system which actually can perform these criteria for an individual cells. First, you can imagine a cellular population where you have the cells of different colors here. Cells have different types of metabolic reactions, different types of cell surface receptor, or it could be different sizes. Some are big cells, some are small cells. So irrespective of that, first you are looking for a system so that it actually can distinguish and analyze the cell mass and give a single cell. And once the single cell is coming out of this tubing, it could be illuminated with the help of laser, and when you illuminate these cells you can able to have the detectors, and these detectors could detect the size of these particular cells, or you can have the detector which detects the density of these cells, or you can have the detector which actually can detect the fluorescence. Now from where you are going to get this fluorescence? For example, if these cells have the different types of receptors, what you can do is you can just add the tagged antibodies which actually going to bind to these particular receptors and is going to give you the fluorescence, right? And that can be detected with these kind of fluorescence detectors. And then at the end, you also require a cell collector. So in case, suppose you want to collect these particular cells for further analysis, then this is actually a funnel kind of thing, and that actually can be used to detect or to collect these cells. So this is a basic instrument design. Now, we can start with the tissue, or we can start from single cells, or we can take any other source of cells like cultured cells, cell line, and bacterial cultures, etc. Whatever source you are starting from, you always have to prepare cell suspension. And this cell suspension will be used as a sample. Now, you put that cell suspension in a flow cytometric cuvette, and you put this cuvette in the flow cytometer. Now flow cytometer will suck up the sample and will start analyzing it. Now we see what happens inside the flow cytometer if we talk about the functionality of the instrument. So very simple, and this is a sample which is a mixture of cell. So when this mixture of cells are going to enter into a tube, it is going to be first focused, then we are going the single cell coming out from this tubing, and then this single cell is going to be illuminated by a laser source. As the cell gets illuminated, it gives signal, and these signals can be detected by the different types of detectors, whether it is the detector for detecting the forward scanning, or it is the detector for side scanning, or it is detector for detecting different types of fluorescent channel, FL1, FL2, or FL3. All these are going to be processed by the processing unit, and then it is going to be converted into a readable signal that actually is going to be analyzed by the computer. So when you see this particular cytometer, this instrument has three basic components. One is fluidics, optic system, and third one is electronic system. So this flow cytometer is made up of three main components: fluidics, optics, and electronics. Fluidic system transports particles in a stream to the laser beam for interrogation. The optic part consists of lasers to illuminate the particles in the sample stream, and optical filters to direct the resulting light signals to the appropriate detectors. And the third part is the electronic system, which converts the detected light signal into electronic signal that can be processed by the computer. So, in the flow system, there is flow cell. The purpose of the flow cell is it is actually going to focus the sample so that there will be single cell at a time. This is focusing of the sample is being done with the help of sheath fluid. So when the sample is injected into the flow cell, this flow cell have the sheath fluid. So there is outer sheath fluid, and this fluid contain 0.9% or PBS, and then it actually encloses the central core. So in the central core, you are going to have the sample which is surrounded by the sheet fluid. Then it is going to use the hydrodynamic process to focus the sample. So let us see what happens in the flow cell. Your sample is going in the form of cell suspension, where you are having so many cells. Now the sample is squeezed by flow of fluid, and this squeezing of sample is called as hydrodynamic focusing. Nowadays there are some instruments which are using both hydrodynamic pressure as well as acoustic waves to do the focusing of the cell. Now what do you mean by focusing? Once you are applying this hydrodynamic pressure on the cell, then cells are coming in the machine as a single array. Thus, flow cytometer creates a condition where you are having one cell at a time passing through a stage where the laser is interacted with that cell. So again you are having your cell in cell suspension. You have created a hydrodynamic focusing. That is the function of the machine, and because of that hydrodynamic focusing, we are having a condition where cells are present in a single array, right? So now these single cells will reach to a stage where laser will put some light on this cell, and the cell will interact with that light, and this interaction is detected by different detectors. Now, the next component is optical system, where you can use light source in flow cytometer. Three types of sources like laser, then arc lamp, and argon lasers can be used of 488 nanometer wavelength. As far as the detectors is concerned, you can have multiple detectors. The detectors which is actually going to detect the forward scanning or, or scattering, or you can have no to the side scattering. So let us see what is this Fs and Ss, that is forward scattering and side scattering. So what happens when a cell interacted with light? So here is a cell. This is laser. The laser is putting incident light beam on the cell, and cell is doing different kind of interaction with this light. The first kind of interaction is the scattering of light at small angles, is between 0.5 degree to 5 degrees, and this scattering of light is called as forward scattered. That is the first parameter of flow cytometric analysis, which gives us the information about size of cell. Now second interaction of light is scattering of light, but at larger angle of 15 degrees to 150 degree, is called side scatter, SSC, which is the second parameter of flow cytometric analysis, which gives us the information about internal granularity, that is complexity of the cell. Now if the cell is having fluorescent factor or fluorescent proteins or fluorescently labeled antibodies bound with specific proteins, they will absorb the light and emit fluorescence, will give information about these molecules. This is the cell, and it is passing through the incident laser light. As a result of this, it will reflect some light in forward direction called forward scatter, and it will be detected. It will be detected um, uh, in photomultiplier tube. Forward scatter light shows cell size, shape, volume. As the forward scattered light increases, this means the shape or size of the cell is big, right? Second one is the side scattered light. There are some granules inside the cell. Some cells have granules and some cells don't have. So how many granules and granular complexity is and how this granular complexity is determined by this side scattered light. This is the Now see, this is the incident light, and it is reflected by the granules of the cell, and it is scattered sidewise. So this light measured at the 90 degrees angle to the excitation light. It is called as side scattered light. Filters and photomultiplier tubes are placed at 90 degrees angle to the incident light, and it detects side scattered light, which indicated cell granularity of of the internal complexity of the cell. Thus, the measurement of FSC and SSC are used for the differentiation of cell types in a heterogeneous cell population. For example, this is the dot plot of the FSC and SSC. In this plot, this is the lymphocytes. Now, lymphocytes having very less internal complexity, you can see here, complexity or granules. So, it will show low SSC, but FSC, that is forward scattered light, it is having size little much bigger, so it shows FSC between 100 to 150. Then you can also you can see here monocytes also. Monocytes are bigger than lymphocytes, so it will show FSC much more than lymphocytes, and it is having granules inside them, so it will show SSC much above the lymphocyte. The neutrophils are bigger than monocytes and lymphocytes. It is showing FSC larger. That means they have larger size, and SSC is also high in neutrophils. That means inner complexity is higher. Then the third component of the flow cytometer is electronic system. Electronic system actually converts photons to photoelectrons. It allows to measure the amplitude, area and width of photoelectron pulse, which means what is the what is the height of the particular signal, then area of the particular signal, then how many signal you are getting. That is actually going to be the purpose of having the electronic system. Okay, so the electronic system is only to take the signal from the optical system, and then it is actually going to convert that to a readable count, and that is going to be visualized by the person who is doing that experiment. So that's all for the flow cytometer design and component. In the next part, we will discuss about how data analysis is done by using this flow cytometry technique. So please like, subscribe and share this video.